Yeah. So greetings from Indonesia. First of all, I would like to say thank you to the organizer of the 48th Global Summit on Biotechnology and Food Science for this platform provided. My name is Semuel Leunufna. I'm from Indonesia, Pattimura University, Maluku. I also represent the Center for the Conservation of Maluku's Biodiversity. And in this opportunity, we'll discuss the topic of cryopreservation and genetic stability on Yams species or Dioscorea spp. So this will be the outline that I'm going to follow through during my presentation. Cryopreservation is a conservation of biological materials under a very low temperature of liquid nitrogen, which is minus 196 degrees Celsius, for indefinite time period without altering genetic constitution of the materials cryopreserved. This method or techniques has been the only method for the establishment of a long-term collection or a base collection for vegetatively propagated crops and crops producing recalcitrant or semi-recalcitrant seeds in an in situ conservation institution or a gene bank institution. This technique has also been found to be very effective in producing virus-free plantlets in vitro culture, even more effective than the meristem culture, which have been widely implemented. It is therefore very useful in the germplasm exchange. It is also have been implemented in the area of cryotherapy. The research on the protocol development the cryopreservation has been started more or less in the middle of 1930s by the use of the leaves of Ligustrum vulgare in the study of George Luyet from England. During this early development, the method used was a slow freezing method or stepwise cooling method or programmable method in which the explants are exposed to stepwise reduction of the temperature down to the minus 40 before plunging into liquid nitrogen, then followed by the rewarming and further cultivation for the evaluation of shoot recovery. The use of the cryopreservation in the routine conservation of plant germplasm, however, started in the early 1990s by the development of what they call the rapid freezing methods or vitrification-based methods, marked by three distinctive methods, which are vitrification, developed in Japan and the USA by the research group of Professor Akira Sakai and Professor Peter Steponkus. The other is the encapsulation dehydration, developed in France, and droplet freezing method developed in Germany. Parallel to the development of the rapid freezing method, the development of the solution called plant vitrification solutions, which is now the PVS 1, 2, and 3, and 4 has been produced and used in different laboratories in the world, was also taking place. The vitrification solutions is the combination of the chemical compounds, which mainly are sucrose, glycerol, ethylene glycol, and dimethyl sulfoxide in certain concentrations. Vitrification itself is defined as the formation of a glass-like structure at a temperature at or below the freezing point of solution. So in the vitrification solutions, the explants, the excised from the source explants, then treated with a certain or cultivated in a certain concentration of medium, then in the pre-culture and treated with the half concentration of PFS2 solutions, which is called loading, followed by the dehydration in the PFS solution, then immersion into liquid nitrogen. Before the explants were then rewarmed and further cultivation for the evaluation of shoot recovery. The encapsulation dehydration method has the distinctive feature, which are the explants are encapsulated in the calcium alginate beads before the dehydration in the desiccator, and followed by immersion into liquid nitrogen, then thawing or rewarming and further cultivation for the evaluation of the shoot recovery. Droplet method has the distinctive features of explants are treated with a droplet of dimethyl superoxide on the pieces of aluminum foil before immersion into liquid nitrogen, followed by thawing and further evaluation for shoot recovery. The recent development in the research of protocol development have been implementing the 3 different vitrification-based methods, and also the combination of the specific feature of these each of these methods. So we will find the methods we called encapsulation vitrification, droplet vitrification, cryoplate or decryoplate, and as well the additional treatment also implemented in the source plantlet before explants are excised for further treatment in the cryopreservation protocol. By the cold acclimations in a continuously lower temperature or alternating temperature, the treatment of the recovered medium is also an area which are being researched in many different laboratories in the world. So in the recent year, the number of the cryopreserved explants in the world have reached about 50,000 accessions, and coming from about 60 species of plants. So beside the in vitro meristems and seeds, which are have been mainly cryopreserved, the other explants such as dormant buds, colors, embryo, and pollen are also being used. Success in protocol development is measured by 2 parameters. First is the recovery of the high percentage of healthy shoot or plants, and secondly, the consistency of the genetic makeup of the cryopreserved materials. In the routine conservation of the gene bank institution, it has been consensus that at least 30% of the shoot recovery can be produced by a protocol, and the protocol can be used for routine conservation of the plant germplasm. With the development of or improvement in the protocol, this percentage probably can be increased. So success in protocol development has been found to be genotypic dependent, which means certain protocol developed for a certain genotype is not necessarily applicable to other genotypes, even within the same species. Protocol validation is necessary for the purpose of meeting for which the accession is cryopreserved. Otherwise, the accession will not be very useful after the very long time of cryopreservation. At the temperature of liquid nitrogen, which is 126 degrees Celsius, the vitrified biological material are said to be in the standstill stage, in which we can there is no biological activities or biochemical activities taking place, and therefore it is guaranteed that there will be no genetic change within the materials. However, confirmation of the protocol is necessary because of the protocol involves different procedures which cause the physical and biochemical stress to the biological materials. So the approach to protocol evaluation and the advantage and disadvantages has been of those approach have been discussed by Professor Keith Harding from England, and molecular marker has been implemented, and it is a reliable marker for DNA fingerprinting. It is usually, however, usually considered only very small fragments of the DNA. RFLP contain about 10 fragments and about 10,000 base pair. Rapid making use of about 15 fragments and 2,000 base pair, or single strand repeats making use of 50 fragments or about 2,000 base pair, and AFLP make use of the less than 1,000 fragments or about 1,000 base pairs. So this fragment is a very small fragment of the whole genome, and therefore the polymorphic DNA fragment is most probably detect the genetic stability rather than genetic instability. The phenotypic evaluation has also been used for the evaluation of the genetic consistency. This phenotype represents the whole genotypic interaction with the environment. However, it also can show the phenotypic plasticity, as well the mutation or deletion at an uncoded DNA area are not detected. Cytological evaluation by determining the chromosome numbers and ploidy changes is also have been implemented. The difficulties is that to observe the number of chromosomes which possess the very small number, very small size of the chromosome, due to the also higher polyploidy in many plants. Biochemical evaluations has also been implemented. Isozyme or a protein is a good indicator of the gene expressions. It is, however, also only a small fraction of the total protein in plants, and therefore it is also usually influenced by the environment. In our study, the Yams or Dioscorea species was used as the experimental materials. Yams has been the staple food in many different regions in the world, specifically countries in Africa as well as in Asia, and it's therefore have been supporting the food security in those regions. Also many species of yams also produce the secondary metabolite or diosgenin, which has been used in the development of steroid drugs. According to Knut, in 1924, there exists about 600 species of yams, fall into 4 genera and 60 sections. Recent report, however, indicating only about 400 species of yams distributed in the tropical, subtropical, and in the temperate area. So the objective of this study is to validate or to evaluate the genetic consistency in yams following cryopreservation by means of in vitro multiplication rate, morphological, and agronomical characters. During our study, we were able to collect more than 70 accessions from the mostly botanical gardens around the world in the Institute for Plant Genetics and Crop Plant Research, IPK Gatersleben, Germany. And we were able to develop the protocols for Dioscorea bolivifera, Dioscorea polystacea and Dioscorea cayennensis lambs. The protocol, the method implemented was droplet vitrification by first the preconditioning the source explants in then alternating cold and warm temperature for certain period of time, followed by pre-culture in high sucrose concentrations, and then loading the explants and to half concentration of the PFS2 solution, dehydration and PFS2, and the cooling or freezing of the explants on the pieces of aluminum foil and droplet of PFS2 solution. Following thawing or rewarming, the explants were unloading and cultivated in a dark period, in the dark for a certain period of time, followed by cultivation in the light. So using the protocol, those protocol developed, we were able to obtain maximum 50% for Dioscorea bulbifera and 40% for Dioscorea polystachya and 30% for Dioscorea cayennensis. The shoot recovery from the cryopreservation of each species were multiplied to produce 18 tubes and then followed by the 3 time subculturing every 2 months for the observation of the in vitro multiplication rate, as well after 4 months of collection, the explants, the survived explants together with the in vitro culture explants were transplanted into the greenhouse and cultivated in pots for the observation of the morphological and agronomical characters. The result of the studies are indicated in the first table here, related to the in vitro multiplication rates. As we can see that for Dioscorea polystachya and Dioscorea cayennensis, the in vitro culture only, in vitro multiplication rate for the treatment in vitro culture only, and are similar in the number in multiplication rates in comparison to the cryoprotectant treated and liquid nitrogen treated or cryopreserved explants. A similar phenomena also seen for Dioscorea cayennensis. For Dioscorea bulbifera, on the other hand, the cryoprotectant treatment and cryopreservation treatment produce significantly higher multiplication rates in comparison to the in vitro culture only explants. So these differences, however, we explain it as the phenotypic plasticity of the in vitro multiplication rate due to stress that is caused by the application of the cryopreservation protocol. And also we conclude that it will be this elasticity will be then returned in the further subculturing cycles. So even though the to have the proof on the consistency of the multiplication rate of the 3 different treatments, we need to cultivate them further, but this was not conducted during the specific study. However, we are making use of the medium-term conservation of the in vitro conservation of Dioscorea, which were done parallel to our study. We can observe that the in vitro multiplication rate only for 3 different or for 4 different species, were studied, and for 3 different species, Dioscorea polystachya, Dioscorea bulbifera, and Dioscorea cayennensis, the multiplication rate was constant in the 10 cycles of subculture or 2 years observation. However, we observe that there are always the fluctuations in the multiplication rate every cycle of subculture, and these also fluctuations are quite pronounced for Yams 16 or Dioscorea bulbifera, as well as for Dioscorea cayennensis. For Dioscorea alata, there is a quadratic regressions that is significant, showing by the in vitro multiplication rate during the 10 cycles of subculture. This means that there were significant reductions of the in vitro multiplication rate in the beginning of the subculturing period. Well, however, the multiplication rate was increased following the further subculture of the explants. And based on this, also we can assume that there will be recovery on the in vitro multiplication rate after the 3 different treatment variants on our study. For the morphological characters, we can see from these pictures, the left side of the pods are the non-cryopreserved explants, but the right sides are the cryopreserved explants. And the observation for morphological differences was done by the use of the Dioscorea descriptor, making use of only a number of characters. And the colors, for example, were found to be similar, making use of the Royal Horticultural Society color chart, as well as the leaves colors, stem colors, and other part of the plants. The tones are produced at the bottom part of the plant and very pronounced at the first cycle of subculture. And this is the cryopreserved, non-cryopreserved, and cryopreserved explants. So they are similar for both treatment variants. This study also specifically done for the Dioscorea cayennensis. Similarity also observed for the stem shape, which is round, as well as the twining of the stems, which is anti-clockwise directions, or from the right to the left. These typical directions for the enantiophyllum sections. For the agronomic traits or yield characters, we can see that the number of tubers are also not significantly different for the non-cryopreserved and cryopreserved plants, pot-grown plants, and in the first cycle, as well as in the second cycles of cultivation. The same phenomenon also seen in the tuber harvest weight, first cycle and second cycle of growth produce no different tuber harvest weight for both in vitro only and cryopreservation pot-grown plants. The pictures are seen in the right side of these slides. I went through the literature on the genetic consistency studies on Dioscorea species, both for the food crops and as well as the medicinal Dioscoreas. We can see that the conclusion shown by all the studies, or about 16 studies, indicating stability of the genetic constitution or stability of the characters evaluated before and after the cryopreservation. So the conclusions can be drawn from these studies. First, application of cryopreservation protocol or procedure on Dioscorea bulbifera caused significant increase in the in vitro multiplication rate. It is, however, considered to be a fluctuation of the phenotypic characters due to stress conditions and will return in further cycles of subculture. And in vitro multiplication rate, morphology, and yield characters evaluated in this study are similar for cryopreserved and non-cryopreserved treatments. As well, the protocol is therefore developed for Dioscorea cayennensis and Dioscorea polystacea and Dioscorea bulbifera is deemed to be valid and therefore can be used for the cryopreservation of the species. This study was done in the laboratory or in vitro storage and cryopreservation laboratory at the Jimban department in the Institute for Plant Genetics and Crop Plant Research in Germany, headed by Dr. Joachim Keller. Some of the technicians and scientists have also very helpful in the supporting their research. The specific study was published in this publication, in this journal, and also some of the references that are used in this presentation. So thank you very much for your kind participation. I would like also to mention Dr. Barbara Reed from the United States Department of Agriculture, and one of the prominent scientists in cryopreservation. Thank you very much.